I'm going to go through the structure and function of different organelles used to synthesize proteins and carbohydrates inside a cell. So the first one I'm going to start with is this one. This is a ribosome. Now, if we think about ribosomes, they're actually too small for us to see on a light microscope, but you can see them on an electron micrograph. But they don't look like you'll see in this image here. When you look at a ribosome on an electron micrograph, it's so tiny that it actually appears as a dot, like you can see appearing here in the bottom right hand side of the screen. Now these would be called free ribosomes. They're going to make proteins for use inside the cell. Also, you will have something called a bound ribosome, which is a ribosome attached to a membrane, like you can see here in the bottom right hand corner. Now these ribosomes are going to make proteins for outside the cell and are attached to the rough endoplasmic reticulum. Ribosomes found in a eukaryotic cell are 80S ribosomes. These are the larger ribosomes that we tend to get. And then ribosomes in a prokaryotic cell are 70S ribosomes. The S isn't coming up very well there, which are the smaller ribosomes that you get in prokaryote cells and also in chloroplast and mitochondria as well. So if we look at the structure of a ribosome in a bit more detail, what we can actually see is it's made of two subunits, one large subunit and one small subunit. Now, with these subunits, they have different attachment sites based on them. So the small subunit has an attachment site for messenger RNA, which would bind onto here. Then the code on the messenger RNA would be read, and it would be joined with complementary anticodons on the transfer RNA at two different attachment sites on the large subunit, and the transfer RNA would bring along with it the specific amino acid needed in order to make that part of the protein. And then these amino acids would all join together with peptide bonds and form what's called a polypeptide, which is the basis of a protein. The polypeptide would then move, for example, in a vesicle to the Golgi apparatus, where it's going to be processed, packaged, and made more complex. What we can see here is the nucleus of the cell up in the top left corner, and we can see the double membrane that surrounds the nucleus, which is the nuclear envelope. Continuous then with that double membrane, here coming out is the rough endoplasmic reticulum with the small black dots on it. Okay, the black dots are, remember, ATS membrane-bound ribosomes, and we can see them just there. This is called rough endoplasmic reticulum. So all of the red with the black dots on it is the aria, and the reason it's labelled rough is just because the small black dots, which make a rough surface when we have a look at it. The whole point of the rough endoplasmic reticulum is basically protein synthesis. So it's where you're going to synthesize but also transport proteins around the cell. Now they're transported through the cisternae. So here is the cisternae. Each of these are flattened sacs with a small space in the middle where any proteins that are synthesized inside the aria can be transported, or any perhaps carbohydrates or lipids synthesized in the smooth ER can be transported as well. So this lower part, the smooth endoplasmic reticulum, is where we synthesize things that are not protein, okay? We know it's smooth because there's no dots on the surface of it, so there's no ribosomes present. So things that are made there, carbohydrates, the basics of cellulose would be made there and go on to be made more complex inside the Golgi apparatus, and lipids, for example, as well, such as steroid-based hormones like estrogen. What we can see here is once the proteins are made, they bud off in little vesicles that are formed to carry the protein to the next part of the process, which is the Golgi apparatus. Also, any lipids, for example, that have been made in the SER as well, or carbohydrates, will go through the same process. They'll bud off, form little vesicles, and again move to the Golgi. Now the next structure, so what we can see in front of us here is the Golgi apparatus. Here at the top we've got some incoming transport vesicles, so they would have come from, depending what they contain in them, the rough endoplasmic reticulum or the smooth endoplasmic reticulum. You can see that the membrane of the transport vesicle here is fusing with the cisternae, which is the same name as we had for the membranes in the RER and the SCR for each of the flattened sacs. Now the Golgi body stands on its own inside a cell, okay? We can see it has this specific shape where it's narrower, gets wider and gets narrower again, okay? It's often surrounded by lots of vesicles because it's got these incoming transport vesicles and then it's got the secretory vesicles at the other side of it. What's the Golgi going to do? It's going to take any of these proteins or carbohydrates that come into it. It's going to process them. It's going to package them, ready to go off somewhere else, either inside the cell or to leave the cell by exocytosis, like we can see down in the bottom right corner. Also, it's going to add to proteins non-protein components. So if you have, for example, a protein and you add a carbohydrate to that protein, you could make a glycoprotein, which can then be put into the cell membrane of the cell, okay? And that would act as maybe a receptor in the cell membrane of the cell. So it's got a lot of really important roles, okay? This is where proteins are made a lot more complex and into their final tertiary structure, which is three-dimensional, before they either leave the cell or they go on to do a different role inside the cell. Okay, so we've got incoming transport vesicles, we've got our proteins being processed, we've got each of these sacs, which are called cisternae, that we can see here, and then here we can see at the end of the process another vesicle budding off. Now this vesicle could have several different names. It is a Golgi vesicle. Any vesicle leaving the Golgi apparatus is a Golgi vesicle. It's going to contain whatever has been produced inside the Golgi body. It's also a secretory vesicle. So for example, this would be a secretory vesicle. It's moving towards the cell membrane. It then fuses with the cell membrane and the contents of it are then excreted by the process of exocytosis to the outside of the cell. However, some vesicles stay inside the cells. So lysosomes are another vesicle that can be produced. Now a lysosome inside its vesicle would contain enzymes. Now these enzymes are hydrolytic. Now what that means is the enzymes are able to break down other molecules by hydrolysis, the addition of water. And they basically stay inside the cell in order to break down worn out and old organelles so that the parts of them can be recycled. Okay, so let's put it all together. So think carefully about all the different organelles together. So we've got the nucleus in this top left corner. Now inside the nucleus you've got the DNA. The DNA is going to be read and it's going to be turned into a code, okay? Now this is called transcription. So the code is going to be a molecule of messenger RNA, if you remember. That messenger RNA is going to leave the nucleus. It's small enough to leave through these tiny little nuclear pores, and it's going to attach to a ribosome. Once it's attached to a ribosome, the ribosome is going to read the code on the messenger RNA, and if you remember, it's going to then translate that code into a specific sequence of amino acids joined together with the help of transfer RNA to make a polypeptide, which is a protein. Then that protein is going to be transported, as you can see here, in a vesicle. So here's a vesicle that could contain the protein inside of it. It's going to be transported and it's going to then fuse with the membrane of the Golgi apparatus, and the protein is going to enter the cisternae of the Golgi apparatus, where it's going to be processed, packaged, perhaps non-protein components are added to it, and then down here it's leaving the Golgi apparatus as a Golgi vesicle, at which point it could move towards and the vesicle could fuse with the cell membrane to release any substance it contains by the active process of exocytosis. Or the vesicle could stay inside the cell. It could be a lysosome, and if you remember, lysosomes are going to fuse with the membrane of an organelle, such as here a mitochondria, and it's going to then release the hydrolytic enzymes into the organelle, which will break down the constituent parts of the organelle by hydrolysis so that that could then be recycled within the cell itself.